So I'm going to talk about the ASAP Consortium, XChem, Diamond Light Source, the COVID moonshot, and how that led to Fragmenstein, and how our current fragment elaboration and hit discovery has got a multi-user angle that we learned from the COVID moonshot, and and this diversity also has a lot of commonality in how visual inspection need for fidelity and the various tools used for that, such as fragalysis. So starting from the top, the ASAP Consortium, there have been several talks here previously that have discussed the ASAP Consortium. ASAP is a backronym of AI-driven structure-enabled antiviral platform. Namely, it stems from the COVID moonshot, and it's taken several lessons from that. Namely, it's collaborative, it's global, and it's antiviral. And several talks have also stressed previously the overlooked viral pathogens. We're not curing the common cold, we're we're developing antivirals against neglected diseases, viral diseases. And there's a variety of methods. The acronym says AI, but we also use molecular mechanics and majorly structure-based. That means high-throughput crystallography, which leads to the coolest toy that you can find in Oxfordshire, namely the synchrotron. This 3/4 of a kilometer wide ring has got loads of beams, and basically, and one of them is Frank von Delft's over here shown sticking his finger in front of the beam, as one does for some reason. And that beamline, the IO41 beamline, is the XChem beamline, does a drug discovery via a high throughput fragment-based approach. And this is possible thanks to better detectors, robotics, and various algorithms operating behind the scenes. It's operated for nearly a decade, but it's not been a static process. It's been continually evolving, Kaizen, as they call it. And basically, this evolution of XChem is actually driven by collaborations. This slide over here is an old slide showing all the various collaborators, and that's just a fraction of potential people that ought to be thanked for various things along the way. That just shows how collaborative, how user-driven XChem facility is. And to show the benefits, it actually speeds stuff up by an order of magnitude, which is awesome. And the secret sauce of it is, in addition to the things mentioned, is fragment-based drug discovery. And fragment-based drug discovery, two principles are levied. One is the ability to detect weak binding. The other one is analogs bind similarly to their parent compounds. And you can merge, link, and expand compounds, even though these compounds may not be the same or synthons based on the others. For example, caffeine, capsaicin, and melatonin are not based on those fragments. And so, yeah, so you would soak 1,000 compounds, you get 100 hits in various spots that have, and one of those pockets might be relevant, and you've got well over a dozen hits generally in the relevant pocket or pockets. So from there you can do all those operations. So that's where the stuff happens. The COVID moonshot used the XChem facility to find inhibitors for the SARS-CoV-2 protease drug campaign, but had an extra spin to it. It was a collaborative, but based on a public call for derivative compounds, and where people submitted a diversity of strategies. And you can read all about it in the paper that came out last month in Science, and which is a great paper, definitely worth to read. Now, one problem that popped up in the COVID moonshot was the fact that a lot of mergers that people were making were based on, say, maximum common substructures or linked in really bizarre ways that did not match the parent hits. Or when enumerating analogs, these derivatives were placed completely independently of the parent hits. There was lack of fidelity. To address this, I created Fragmenstein, which merges fragments by obeying strictly the positional overlaps, and places compounds by docking them. In the case over here, you've got, it might place as if a tautomer were the better choice, for example. So, and together these form the Fragmenstein pipeline, as I call it, where you merge a compound, find an analog by catalog, as I'll go into a minute, and you can, then you've got your compound that you can purchase. The first step on Fragmenstein is the combination, where you've got two hits that you want to combine. They might be imperfect hits. They might not fully overlap. And you've got red herring groups as the ones added, as that methyl group sticking out over there. But when you crystallize those two, the derivative will obey the parent hits, and so, or to some extent, and that's where the energy minimization comes in, as I'll get to in a minute. But to determine which compounds match to which, there's obviously various rules to determine the overlap, and one of the tricks that it does is it collapses rings into a placeholder atom. As a result, that allows a 5-membered ring and a 6-membered ring to be merged very nicely. And at the bottom of this picture over here, there's a furan and a benzene that are being overlapped and translated. And the more they get translated along, the more different compounds appear. When the oxygen's hidden, you would get a naphthalene or a benzofuran on the other side, a spiro, and then it forms links. Fragmenstein doesn't do links properly. It just adds a hydrocarbon chain of the ideal length. We saw previously in the seminar series, Steph Wills gave a great presentation about a different method that allows novel linkers to be enumerated. However, once this stitched together monster is created, we do have a problem that the chemistry might be wrong. For example, if you merge two oxygen, if you got a tetravalent oxygen, that is not perfect. So as a result, there's some chemistry correction that happen. And actually, this has found a new home in diffusion models where the de novo compounds are definitely not what you'd hope them to be. So it does require chemistry sanitization. And analog searches are required with Fragmenstein, as I mentioned, to find a compound that you can actually purchase from catalog. And that's where the placement part comes in, that given a smile string, place the smile string based on the parent hits that you want, that you've provided. So this is a traveling salesman problem. You've got various ways to address it. The main way that Fragmenstein addresses it is the second route illustrated here. You've got strict maximum common substructure search followed by a laxer one where you have expand around them more and more and more with a cheat to allow for red herrings to be discarded. However, these stitched together monsters are not actually, they might have some really nasty chemistry, so it might be distorted. So it's important to energy minimize, and Igor is the energy minimizer for that uses Pyrosetta. Other modes that Fragmenstein can use is using RD kits, MMFF, force fields, or OpenFF. A nice example there of Fragmenstein in use, we saw previously, I believe in February, Jamie Fraser presented the merger of two compounds in the MAC1 domain, where these two compounds were merged, and in a single round, the submicromolar IC50 was found, which is very impressive. Although I must say that we're a bit fortunate. There's a bit of a fortuitous event happening there where a Urado group was added. So Fragmenstein can't claim credit for that. That was the catalog expansion that gave an extra group. So current work. So how does this factor into the ASAP Discovery program? So during the in the ASAP program, there's various various targets are screened by the XChem facility, and these fragment hits are then expanded expanded into derivatives, and I'm involved in that, a cog in a greater wheel in that. So I should say that there's quite a big difference with how it's normally done academically. Namely, academically, you've got a single person that's enumerating the compounds, is reviewing them, purchasing them in a relaxed manner. However, we took several lessons from the COVID moonshot where multiple users and multiple techniques are entering play. And the users that create that data are not the people reviewing them. Instead, there's an external to them step, the med chemist reviewer. And a fast turnaround is imposed to avoid dilly-dallying and taking it easy and like one last experiment over there. So strict timelines, and there's a staggered cycle. So compounds are enumerated based on hits that were determined from derivatives, not from the previous cycle, but 2 cycles back. So it's a bit of a staggered process going on. So now let's look at the review process. I said that the compounds submitted are reviewed, and they're reviewed in Fragalysis. Fragalysis was designed by many people in XChem, Informatics Matters, and M2M Solution. I apologize that I can't thank them all. And it's linked to the XChem stack, and it shows on the left-hand side, which is collapsed here, the hits, the fragment hits, and on the right-hand side, the virtual derivatives that a user has submitted. And in this case, there's we can see overlaid. We can in the thicker sticks the parent hits, whereas the thinner ones the derivative. Yes, to use XChem parlance, the inspirations and the follow-up. Yeah, so the med chemists look at this and review and veto whatever compounds seem wrong and such. And so, and that to the methods, the multiple methods are used for that. For now, there's still the human step, which is essential. The PD, I look at the PDB, read some literature in PubMed, generate some AlphaFold 2 complexes to test hypotheses, understand the problems that may help, and tell the others and such. But moving forward, the more the methods develop, the less the humans will have to be involved, which is the aim. So so yeah, there's 3 different course general approaches. We've got shape and color approaches, whereas pharmacophores and the shape of the compound selected for the library enumeration selects on those. Direct catalog enumeration, as in one enumerates all the matches in the catalog for certain criteria and places those, docs those, or de novo generation via a variety of methods I'll get to in a minute, and searches analog, then jumps into the catalogs by an analog search. So starting with de novo, which is my main interest, there's Fragmenstein proper, as I said, which I run, and doing merges, searching for analogs using NextMove software SmallWorld, which is hosted in the University of San Francisco servers by John Irwin and Brian Shoykett. I place those and rank them via various means that I'll get to in a minute. Then another method, the other methods we also use, there's de novo compound generation via AI. Lucy runs Strife to enumerate de novo, to generate de novo compounds. Yanka Sheen, use Silver to enumerate compounds, and these were then slightly corrected for their chemistry, SmallWorld search, and then placed relative to where they are. The analogs placed relative to the inspiration, the original generated compound. One method that was also used, for example, was CSAR F trees by Charlie Tomlinson at one point, and then analog searches for those were placed, and we're continually adding new methods. For example, the new iteration, I'm hoping that there'll be a new conditional equivariant diffusion model present, and then in terms of the other approach, enumerative directly, Steph, as we saw last month, I think, uses a different method than Fragmenstein. Namely, she enumerates all the compounds from a catalog that match certain criteria using shape and color, a shape and color search, or similarity, a fingerprint similarity search, and then those merges, and then enumerates those and places them with Fragmenstein. Then we're very fortuitous in the ASAP project that OpenEye gave us licenses to use their software, and big fan of the results of OpenEye Rocks, shape and color search, and I enumerate compounds from those, and I place them with Fragmenstein for corrections and all. And recently, we've been joined in the process by Josh Horton and Daniel Cole, where they brought FEGROW into the fold, which enumerates expansions and scores them by FEP using OpenMM. And I've been playing around with other methods, developing new methods. Like using the NextMove software's Alpha server, which allows smart searches to be done. And so I've been generating flexible smarts patterns that address several specific hypotheses, such as linkers with different groups and whatnot. And obviously, there's been other approaches such as manual designs, or using the Fragalysis vector expansion, which is a feature in the Fragalysis server. And in future, I'm currently working on another method that hopefully I'll be using at the next iteration. So I've mentioned several people, and I ought to really mention other people involved, because I'm just a small cog in a greater machinery. I mean, the med chemists, as I mentioned, play an instrumental role because they review the compounds they're generated. Ed Griffin, Tryphon Zagaris, Thomas Vargo. And these compounds, once they're shortlisted, they get ordered by Beth McLean, and then they get uploaded to Postera, which I have to thank Jenke and Hugo for helping me deal with formalizing what keys to use. And overall, though, getting all these cogs to work requires a lot of coordination. So that's the work of Max Winniken, Warren Thompson, and Kate Feisler. But the biggest thanks of all goes to Ryan Lithgow and Darren Fearon, who did the who did the wet lab experiments. That is crucial, that without any of this, there'll be none of this. And actually, they've been working incessantly, like insane amounts. So thank you. So yeah. And so that's how it goes. And I should say a small parenthesis on the various methods. Each designer performs their own clustering and ranking of the compounds. Obviously, a method on the paper that gets published in just says, oh yeah, you sort by energy or whatnot, but that's a terrible thing. You're not meant to do that. Instead, there's a variety of things that get done. For example, I cluster by probability scaled interactions and rank them via weighted sum of multiple factors. For example, rotatable bonds, which are not factored in on my energy function I'm using because it's a static snapshot. Rigidifying a rotatable bond is an entropic penalty. So I'm factoring that in my scoring metric to rank them. And yeah, and a curious thing about having an external review step, which is actually really useful, because as by myself, I'm quite biased. I spent a lot of effort on method X. I'm attached to a certain method. I quite like a compound. But by having an external review step, it completely detaches this quite brutally. So it's, I mean, it is obviously sad seeing a compound I like not make the cut, but that's the way. So yeah, so unbiased is that if the two reviewers, one of the two reviewers vetoes a compound, it's a no-go, for example. That's good. An interesting extra thing, for example, that happened recently that I hadn't really thought of, for example, was the virtual compounds are generated by induced fit, but they're viewed in a system where the protein's frozen, it's fixed, it's a template, it's lock and key. So you see clashes, which as a designer, when I look at that, I'm like, oh, my tool is awesome. It's the GUI's fault, it's fine. But when the reviewer sees it, obviously that compound gets chucked up. So there was post-ranking correction to filter out, to correct and filter out the compounds that don't match a lock and key hypothesis, which I thought was very interesting and I hadn't considered. So hopefully, to conclude, I'd like to mention, it was a talk in two parts. In the first part, I talked about Fragmenstein, which is trying its best to be faithful to its inspirations and can work with more than one inspiration molecule and can deal with covalence, is open source, is user interaction driven. I mean, it does have its cons. For example, the main mode runs on Pyrosetta, and it's not super fast. Running without Pyrosetta makes it a fraction of a second, but less precise. And the mergers are not perfect all the time. And in terms of the ASAP hits discovery, as we're currently doing, the key take-home message is that we're using multiple methods, and these methods are getting coalesced into a review process, which is, interestingly, we're using a very accelerated timeline in order to get stuff going, as opposed to dilly-dallying on, one last experiment kind of thing. So very strict timelines, which I would say is very unique. So yeah, and so thank you for listening, and I would like to thank several people that are involved in the creation of Fragalysis, the Fragmenstein, the first part of my talk, and and thank you for listening.